Hey, Sister Mayo, this is our video for lesson 10. Okay, so for problem 1, we drew it up here, but we drew number 4 up here also. So there's a picture of it in the Word document that we'll submit, but just trying to get us to realize where the mRNA fits and how everything is arranged during transcription. So it says, assume that well, suppose a eukaryotic messenger RNA has the following nucleotide sequence, and then it gives a sequence. Assume that RNA polymerase II produced this molecule from right to left. Which end of the mRNA molecule is the 5' end and which is the 3' end? Since it says that it produced it right to left, we said that the A on the right side, that's the 5' end, and then the U on the left side is the 3' end. And B, what is the sequence of the template strand, or the template DNA strand? And the polarity of that's going to be opposite, so the 5' is going to be on the left and the 3' is going to be on the right. And wherever there is, it's just going to be the complementary base pairs. But understanding that whenever there was an A in the RNA sequence, it's going to correspond to a thymine in the DNA sequence. So I guess I could read the letters for you. B is 5' ACGAA. Oh, yeah. ACGAA, CGGTTT, AGTA, GGCT, and then 3'. And then for C, we might have, we might have cheated a little bit for C, but I don't really think it's cheating. We just copy and pasted the RNA, the mRNA, and then wherever there was a U, we changed it out for a T, because the coding strand is the exact, it looks exactly like the mRNA, except for the fact that there's no uracil in DNA, it's thymine. And then you wanted us to draw it. I guess I could show you on the picture we took. Let me make sure that you can actually see it. So this is just the drawing of it, and then that green enzyme on the left was the RNA polymerase II, and that should make sense. And I think that's it for question 1. Yeah.
All right, so for question 2, we have the TAT, a binding protein within the TF2D binds directly to the TATA box, while sigma factor binds directly to the -10 and -35 sequences. And then B is the guanyl transferase, is a eukaryotic enzyme that links the 5' carbon to the 1 nucleotide of the 5' carbon of another nucleotide. C is a group 1 introns ribozymes use the nucleoside guanosine as a cofactor during catalysis, while group 2 introns use adenine as a cofactor during catalysis. And then D is the cleavage and polyadenylation specificity factor, is a endonuclease that assists in terminating transcription in eukaryotes. E, in lesson 9, we learned about the general transcription factors. TF2B is a GTF, which acts as a bridge subunit to link TF2D and TF2F / RNA polymerase II, and TF2H is a GTF, which acts as a phosphorylate and activate RNA polymerase II, and F The spliceosome is composed of subunits called small nuclear ribonucleic proteins. And these spliceosomes subunits are composed of two components, proteins and small nuclear ribonucleic acids.
All right, for number 3, I kind of had a hard time with that first one, but we thought it'd be T2 or TF2H and RNA polymerase 2. For B, the one protein that acts as a helicase to separate RNA from DNA would be a Rho protein. For C, indicate the 3 molecules in eukaryotes that function during RNA modification as ribozymes. That'd be group 1 introns, group 2 introns, and RNAs. For eukaryotes acts as a transcription factor. That'd be sigma factor. And then one enzyme in eukaryotes that synthesizes a long string of identical RNA nucleotides, but does not use a template DNA strand for RNA synthesis. That was the CPSF, which is the cleavage and polyadenylation specificity factor.
Okay, and then for question 4, so we did the first one. We did A, B, and C, and then we didn't have enough room for D, so we erased A. But we did take a picture of it, so you'll have it on our Word document, and I have a picture of it right here. So I'll just kind of talk through it. Yeah, it's okay. So A was kind of the easiest one. We're trying to, this question, we're trying to figure out, we have a hypothesis that there's 4 introns in our favorite gene. And so we're going to use electron microscopy to see if that's true. And so in part A, we take the pre-mRNA. So this mRNA doesn't have any of the introns that have been spliced out of it. So it's everything's in there. We take the pre-mRNA for our favorite gene, hybridize it to the genomic DNA copy of the YFG. And so what that will look like is you'll have the DNA strand running, the template DNA strand we have running on the bottom 5 to 3, and then the pre-mRNA is right on top of it, and it has the introns, the exons and introns, and there's 5 exons and 4 introns for our gene, and so the template strand is totally straight and hydrogen bonded to that pre-mRNA. Now the coding strand forms an R loop that is the length of the pre-mRNA, because all of the template strand is bonded to the pre-mRNA, and so none of the coding strand is bonded in that section to the template strand. And then for part B, it's the mature mRNA is hybridized to the genomic DNA, and so let me make sure you can see from the video. Yeah, you can kind of see. So the way we figured it, this is the mature RNA. So this is the 5' end, 3' end, and we have exon with no introns in between. But the template DNA strand running anti-parallel would still contain those intron sequences. And so, but the exon sequences would be hybridized to the mature mRNA. So the exons are bonded, but you have this intron loop where the intron is for the DNA strand. And then right when we get back to the exon sequence, again, it's hydrogen bonded to the mature mRNA. And since there's nothing wrong with any of the introns in the mature mRNA, there's going to be 4 intron loops because all of them have been processed out of the mature mRNA. Now for the coding strand, there's gonna be an R loop wherever the exons are bonded or wherever they're hybridized. But right when it gets to the intron, the coding strand is going to form a double helix with the template strand until we get back to the exon again. And so we're going to have a couple of R loops from the coding strand and 4 intron loops because all 4 introns have been excised. Now for C, that was a little bit different. It says that the mature mRNA is hybridized, but in this one, the 5-intron in the pre-mRNA contains a defective donor site and branch site adenine. So that intron is not going to be excised. And so if you see our mRNA here, we have a 3' 5' end. The intron closest to the 5' end, it remains. And so that's going to result in 3 intron loops where the template strand loops where the introns have been excised from the mature mRNA, and the coding strand also will form that double helix at those intron loops. And there will be one less, or not one less, there will be 3 R loops. How do I say this? 3 intron or exon R loops. And then once we get past exon 3, there's not going to be any more intron loops because the intron is still in this mRNA, just like in the first one. So the template strand is going to be bonded at this location. And so this coding strand is going to have to wait till the mRNA sequence is done, then it can rebond to the template strand. And then D, the mature mRNA for YFG is hybridized to the genomic DNA copy of YFG. In this case, the 2nd and 4th introns in the pre-mRNA in the 5 to 3 direction both contain defective donor sites and branch site adenine. Assume that the introns are removed using the spliceosome method. So intron 2 from the 5' end and intron 4 from the 5' end were not taken out during processing. And so the template strand is bonded to the mRNA wherever they match up still. So like this exon matches still, but the intron number 1 from the 5' end was taken out, so we have an intron loop. Exon 2 matches up, intron 2 matches up because it failed to be processed out. Exon 3 matches up, but then intron 3 was taken out, and that's just how the rest of it worked. And I think that's it for this question.
Awesome. So for the question number 5, we're talking about the differences of what's mostly found in for the first two questions is eukaryotic versus prokaryotic. And so for A, Rhizobacterium, so it's a prokaryote. And so in that we'd have the RUT, the -35, and the NUSA. For the yeast, which is a eukaryote, it would have the -25 enhancer, group 1 intron, GC box, TF2D, SNRNP, and silencer. And then for C, it's asking which ones are involved in transcription initiation. And that would be -25 enhancer, -35, the GC box, the TF2D, and the silencer. Which of the above is involved in transcription termination? That'd be the RUT and the NUSA. And then which of the above is a DNA sequence? So we kind of struggled with the wording of this. What is a DNA sequence? So we were looking at the -25, -35. They're not necessarily a sequence, but they're locations for the promoter DNA, for the promoter. And so we kind of made a little note of that. But the rest of them that are definitely a DNA sequence would be the RUT, the GC box, enhancer, silencer, and group 1 intron.
Sweet. Have fun on your camping trip.